Good afternoon, everyone. It's a pleasure to be here to introduce the next speaker. My name is Guillermo Amir. I'm the director of the Center for Advanced Regenerative Engineering and a professor in the Department of Biomedical Engineering and Surgery here at Northwestern. And it's my pleasure to introduce Professor David Mooney, a friend and colleague and definitely a role model. We're definitely in for a treat today. Dr. Mooney is the Robert P. Pinkas Family Professor of Bioengineering in the Harvard School of Engineering and Applied Sciences, and a core faculty member of the Wyss Institute at Harvard. He's a leader in the fields of biomaterials, mechanotransduction, drug delivery, tissue engineering, and immunogenic. Born in Madison, Wisconsin, Dave received his BS in chemical engineering from the University of Wisconsin, Madison, and his Ph.D. in chemical engineering from MIT, under the mentorship of Professor Robert Langer. In fact, we both overlapped very briefly while at Bob's lab. He completed his postdoctoral studies at Harvard Medical School and was on the faculty of the University of Michigan before joining Harvard University, where he's currently at. Dave is interested in understanding how cells sense signals in their environment and how these signals alter their behavior. His laboratory developed biomaterials that exploit these signals to regulate specific cells and their functions. They were the first to demonstrate in a three-dimensional culture that the mechanical properties of a substrate can regulate stem cell fate. His lab also developed the first implantable biomaterial cancer vaccine that contained biochemical cues that would re-educate the immune system to destroy cancer cells. Dave's goal is to use our knowledge of cell biology in conjunction with materials to boost therapeutic effects. He is a member of the National Academy of Engineering, the National Academy of Medicine, and a fellow of the National Academy of Inventors. And he's won numerous awards, too many to go through all of them today, but his research and most important, his mentorship and teaching has been impactful. He has published over 400 articles, has several patents in the area of bioengineering, several of which have led to successful commercial products. He's an active member of the major engineering professional societies and an editorial advisor to multiple journals and serves on the industrial advisory boards and government advisory boards. So please join me to welcome Professor David Mooney and his presentation entitled Biomaterials to Reconstitute Cell Immunity. Thank you, Dave.
Thank you. Well, thank you very much for the introduction, Guillermo. And I want to thank Chad and the other organizers for the invitation. It's really an honor to be here, and I'm humbled to be on the same stage today with all these other speakers. I've just really enjoyed the morning, and I'm looking forward to hearing more this afternoon from some of the other speakers. I'm going to talk today about just one aspect of the research in the laboratory, and that relates to the use of materials to try to build T-cell immunity, and I'll talk about this specifically in the context of cancer. Now, this picture that you see in the opening here actually demonstrates pretty much everything I'm going to talk about today. So we develop biomaterials in the lab, and you can see these particles, these microparticles in the background. And the premise is we put information into those biomaterials that we'd like to transfer to cells. And then instead of trying to get those materials to somehow find the cells of interest, instead we bring the cells in the body or outside the body to the material. And you can see cells invading into this material in the bottom part of this panel. And so the premise is by bringing the cells into very close proximity to the material, we can very efficiently download cues and information to those cells and change their behavior. And as I already mentioned, I'll be talking about this today specifically in the context of T-cell immunity and cancer, and I'm going to talk about a specific type of T-cell called the cytotoxic T-cell. And for those of you who are not familiar with this, this little video gives you a sense of what these cells do. These are cells, part of our immune system, that traffic throughout the body, and they're constantly surveilling their environment. If they encounter signatures of things to which they've been trained to recognize. So for example, if these T cells that you see here in red encounter cancerous cells in a tumor, they have the capacity of killing those cells and eliminating the tumor. So because of this capacity for these cytotoxic T cells, they've been the focus of efforts in cancer immunotherapy. And so I'm going to talk to you a little bit to start with now about how these cells are generated and where the field is today. and that'll describe our work to try to manipulate these cells in vitro and in vivo. Now, as the starting point, an understanding of how the immune system interacts with cancer has been popularized by Ira Mellman, as shown by this schematic here on this slide. And the basic premise for how cancer cells interact with our immune system is that cancerous cells have mutations. That's why they're cancerous. And so they make different kinds of molecules like proteins than normal cells. Now, when these cancer cells die, as cells just naturally turn over, their contents will be released into the environment. And there is a type of immune cell called an antigen-presenting cell or dendritic cell. And you can see basically those cells in this first part of the schematic. They constantly are patrolling around the body, and they'll pick up some fragments of these mutated proteins, for example. They will traffic these to the lymph nodes, where they will interact with these T cells that I just introduced you to. They will then present these signatures of the cancer to the T cells, and they can then generate specific T cell responses against these mutations. Those T cells then circulate throughout the body, as I just showed you, and the premise is when they find cancer, they can kill those cancerous cells and clear the tumor. Now, obviously, this does not routinely work in the body, or we wouldn't have so many people dying of cancer all the time. So that's led a lot of research into trying to understand where this cycle breaks down and why it doesn't work. And one of the places that it breaks down is in the tumor microenvironment, where the cancerous cells have evolved mechanisms to specifically downregulate these T cells and keep them from doing their job. Now, based on the recognition of the molecular pathways responsible for this, this has led to new types of therapies that many of you may have heard of. That's been a really exciting development over about the last 10 years in cancer immunotherapy called checkpoint blockade therapy. And the idea of this therapy is that they're typically antibodies that are given to patients, and they stop the interactions of the cancerous cells with the T cells that turns the T cells off. So basically, it lets T cells do their job. And really, the single piece of data that got this field afire about 10 years ago is shown here. And this was the results of the first large-stage clinical trial in the context of end-stage melanoma. This was a trial run by Steve Hody at the Dana-Farber Cancer Institute, and it shows survival rates for patients with melanoma. At that point in time, melanoma was considered to be an incurable disease, and you would ultimately would die from this disease. And so the really exciting thing about this clinical trial is that you appreciate there's this tail on the curve where about 20-25% of the patients went on to become long-term survivors. In essence, they were cured of what was considered to be an incurable type of cancer. Now, since this data was published, there's been a number of these drugs that have been approved by the FDA. They've been demonstrated to have utility in many types of cancer, and it's really revolutionized cancer therapy in the United States and worldwide. So that's really fantastic news. The limitation, though, as you can probably appreciate from this early data, is that only a subset of patients actually benefit from this type of cancer immunotherapy. The majority of patients still don't get benefit. Now, what's been learned mainly from clinical studies is there's a very strong correlation between the success of these therapies and a pre-existing T cell response in the patients. And that really makes sense if you think about it, that this can only work if there actually are already T cells there that you can take the brakes off and have them work effectively. And when those T cells don't exist, then those therapies don't work. So this has led to the idea that maybe what we really need to do is combine these checkpoint blockade therapies with therapeutic vaccines that act earlier in the cycle and amplify the generation of T cell responses. And the premise is if you can generate lots of cancer-specific T cells, you could then combine that therapy with checkpoint, and a very large fraction, maybe someday all cancer patients, could actually benefit from these therapies. So this is what I'd like to talk to you about for the next few minutes, is our activities to try to create these types of what are called therapeutic vaccines, because we're treating patients that already have cancer. Now, about 10 years ago, we first published the concept that underlies our strategy, and it's shown schematically here. And the basic premise is really straightforward. We want to use a material to educate immune cells in the body to be able to skip ex vivo manipulation as much as possible. And so the material is designed, so after placement in the body, the first thing it does is it releases what I'm generically calling a recruiting factor, which then will diffuse into the surrounding tissue, create a concentration gradient that will then bring in the target immune cell. In this case, it's immature dendritic cells, these antigen-presenting cells. Now, once they're inside the device, we then are creating a new microenvironment for these cells that's distinct from all the stimulatory signals the tumor might be letting out to try to prevent this from happening. And now we can very effectively activate these cells with what I'm generically calling programming factors. In this case, we induce a transition of these cells from an immature state to a highly active state that can be very effective at generating immune responses. And then in the third step, we release these cells from the device, they traffic to the lymph node, they interact with T cells, and hopefully trigger this whole process that I've been describing. So that's the premise. Now, our first generation approach to accomplish this is shown here. We decided to take the most kind of vanilla, bland, simple approach you could imagine to try to enhance translation. So we went as the backbone for the strategy to the polylactide coglycolide polymers, whose structure is shown here. These are the same polymers that are used to make biodegradable sutures. So there's a long track record of using these in humans. And we've used a high pressure carbon dioxide gas filming process that I won't go into the details to form basically little tablets that you can see in the upper right hand side. So those are highly porous, about 97-98% space, and those holes are about 100 microns. And we want them to be filled with holes so cells can easily crawl in and we can house a substantial number of these immune cells at any given point in time. Now, to get the cells to actually do this, to get them to crawl in and live inside the device, we exploit a FDA-approved drug under the trade name of Leucine. It's GM-CSF. That's already approved for other indications, but we knew when we started that one of its activities is it would actually attract these cells that we're interested in. So we would encapsulate this into the device, and then to provide an activation for these cells, we would take small adjuvants, small fragments of DNA that would be representative of bacterial or viral DNA, not mammalian DNA, because we know these very effectively activate immune cells. And we would combine these with a polycation to create nanoparticles. And you can see with this TEM, the nanoparticle is about 40 nanometers in size. And we just decorate the polymer surface with these nanoparticles. So the premise is the cells are induced to crawl in. They can then more or less scrape some of these nanoparticles off the material, and they can very effectively become activated. Now, the last component of the system is what we need to give the cells so they can actually recognize cancer. And how we accomplish this is by taking a biopsy of the tumor, turning it into a freeze-dried mixture, and we encapsulate that as well. So we'll have a sustained release of these mutated proteins that can then hopefully generate a specific immune response against cancer. So that was all the components. Now, the first piece of data, again about 10 years old, that we generated that demonstrated this potentially could be useful was when a therapeutic rodent study using the standard preclinical model, where we in essence gave these animals melanoma, and once they had tumors growing in them, we then looked to see whether by vaccinating them, could we basically reverse the cancer formation. And what you can appreciate from these graphs of tumor size and survival is by vaccinating these animals twice with this little piece of plastic, we could get the cancers to slow their growth dramatically, and in over half the animals, we saw complete regression of melanoma. When we published this, this was an unprecedented finding. No one had been able to demonstrate these kind of robust immune responses in this very difficult model. So this got us pretty excited, and we spent then a number of years doing a lot of stuff that I won't describe here to move this forward to actually be a human therapy that we call WDVax. And this is an image of what one of these vaccines looks like, held between a pair of forceps. We moved on and have fairly recently completed a first phase 1 clinical trial in terminal stage melanoma patients. And this is actually a picture of Steve Hody, who's running this clinical trial. And next to him is Mary Gooding, one of the first patients who was treated with this therapy. Now, we're currently finishing the evaluation of all the data from this study. Steve has not yet released it, and because I have a conflict of interest, until he makes the data public, I don't have the data. So I can't tell you much more about this, except that it appears that we were able to achieve similar types of responses, at least grossly, in these human patients as we did in the mice. So we're eagerly looking forward to moving this therapy forward. Now, this initial trial was an academic trial, something that we actually did ourselves. But clearly, we're not going to be able to go on to do large-scale clinical trials ourselves. So we recently licensed this technology to Novartis, the pharmaceutical company, and they are currently commercializing this therapy, hopefully for much broader use. Now, while we're waiting for that to happen though, we're not just kind of sitting back and waiting to see what happens. We recognize that there are a number of limitations in the system that I've just briefly described to you. One of the limitations is that we have to actually do surgery on a patient, make a small incision, and place this disc that's about the size of a baby aspirin tablet under their skin. And that works great in Dana-Farber Cancer Institute in Boston. It's not going to be an optimal situation in a lot of the places where many patients receive care. We'd like to move this to an injectable system. And also, we couldn't actually make these devices until we did the biopsy from the patient and then turned it into this freeze-dried mixture of antigens. So there was a very significant delay between obtaining the biopsy and treating the patients because we had to make the devices. So what we are doing in the next generation system is to move to a needle injectable system that can be introduced in a minimally invasive manner and where we can prefabricate the entire thing except for the antigen. And once we have the antigen, we can simply absorb it onto the particles and be ready to go. And these antigens may either be common antigens that are shared by many types of cancer or patient-specific antigens like I've shown you in this last slide. Just to give you a sense of how this works, the basic premise here is we use microparticles of mesoporous silica. And by having these long aligned nanopores inside these particles, that provides a really ideal location to park the biological cargo. These molecules we use to bring in the cells and the nanoparticles we use to then activate them. Now, these microparticles can be suspended in saline and injected via a needle, but once they're actually in the body, the saline dissipates in the surrounding tissue, the particles collapse and form a three-dimensional porous material. So in essence, we're creating a scaffolding in situ instead of having to preform it outside the body and then introduce it. And once it's in place in the body, all the same biology that I've already been describing to you then happens, where we bring in the cells, we activate the cells, we give them antigen. And I should mention this work is all being done, or was being done by Eileen Lee, who recently left the lab for a postdoctoral fellowship, and Jae-Yoon Kim, who's now running his old lab back in South Korea. This is what these particles look like. You can see they're about 80 to 120 microns in length. And then on the right, you can see a TEM of these long aligned nanopores, about 7 nanometers in size, where we park the biological cargo. Now, when we inject these, you do form a small nodule under the skin, as you can see in this mouse. And there's some histology there showing you that we have attracted large numbers of immune cells. And what this video shows you is particles that were labeled in green. And now we're doing confocal through the tissue that we've sampled. And you can appreciate that we have a lot of cells in orange that have now infiltrated between all these particles. Now, I must admit, after this morning's talk, I feel a little bit foolish presenting confocal. Because I'm sure Professor Hell, who's out there right now, is feeling sorry for me right now. But anyways, we can bring in the cells. As I mentioned, we can coat these very readily. We actually coat the particles with a polymer called polyethyleneamine. And what that enables us to do is to take random mixtures of antigens and readily absorb them. In this case, we're just looking at absorption of particles either with or without the coating. And these are a panel of what are called neoantigens that I'll talk more about in a minute from actually mouse cancers, and on the right, human cancers that we've obtained those from Kathy Wu, who's currently running a variety of what are called neoantigen clinical trials at the Dana-Farber. So it looks like the system can be broadly useful with a wide variety of different types of materials. And just to give you a sense of the power of this system, this just shows you another type of preclinical study where this is a different types of cancer that we're looking at treating. In this case, we allowed tumors to form. We injected at the start of these graphs you see on the lower left. In the controls, basically those tumors grow exponentially. The animals go on to die pretty quickly. And we have 2 flavors of the vaccine. The optimized one is the VP here. And what you can see is with that optimized vaccine, we get complete regression from the vast majority of the animals of the pre-established tumors, and about 90% of them go on to be long-term survivors. So this general strategy seems to be very effective even as we change material systems. Now, I've mentioned to you several times that we want to generate T cells, but I actually haven't shown you that we generate T cells. So just to give you a sense that there is some data behind these pretty pictures, this is just some numbers of T cells that we generate that are specific for these mutations that we deliver. In this case, it's just a couple different ways of measuring these specific T cells, but what all these have in common is in the N animals, the naive animals that we've not treated, there's pretty much no T cell response. And in the VP, which is the optimized vaccine, we generate really robust increases in the number of cancer-specific T cells in these animals. Now, to kind of go back to where I started with cancer immunotherapy, I'd mentioned earlier about the idea of checkpoint, and that checkpoint therapy hopefully would be synergistic with these types of therapeutic vaccines. And what Eileen is showing here is that indeed, if we look at the effectiveness of one of these checkpoint therapies in this model, and this is the anti-CTLA-4 condition in the middle, it has a very modest effect, but if we combine checkpoint with these types of therapeutic vaccines, we get really robust immune responses and really robust effects on established cancer. So it does look like there is a really robust synergy preclinically. The next clinical trial is established or is intended to establish whether or not this also happens in vivo in patients. Okay, so that gives you a little flavor of some of the things we've been doing. I'd like to spend a few minutes talking about what we're doing right now that's not published. And one of the challenges we have, I've mentioned antigens a number of times. Kind of the flavor of the day and really the excitement in cancer in terms of identifying things to target T cells against is to actually sequence each patient's cancer. So you obtain a piece of tissue, healthy tissue, a piece of cancerous tissue, you sequence it, you use these really complex algorithms that I don't really understand, and you use those to predict what mutations are likely to lead to antigens that can generate strong immune responses. And then you synthesize the peptides, and then you put them into systems like the ones I just described. Now, if you think about this, this is a really long process. It's a really expensive process. Actually, our ability to predict is not that good, so it has a relatively low success rate, and a lot of them probably are not going to work all that well. So right now what happens, you just make as many of these as you can and kind of hope for the best. Now, what we were very interested in is the idea that perhaps we could vaccinate without an antigen. And the initial reviewers didn't like this term, but I think it really is accurate. So Nisarg Shah, who recently moved to UCSD, is the one who was leading this work before he went on to start his own laboratory. And he's been exploring this in the context of blood cancers, and particularly acute myeloid leukemia, which is a disorder of blood stem and progenitor cells. As many of you may know, that the typical therapy for these patients is to get cytotoxic chemotherapy, and it actually works quite well and causes regression, but then the vast majority of those patients will end up relapsing. And then the question becomes, how do you actually treat them after they've had this relapse? So as a first step to this, what Nisarg basically decided to do was develop a material system that we could use. And I'm not going to go into all the background, but he's explored a different system for a little bit, for some specific reasons. But this is a macroporous material. Here again is a confocal image just looking through the material. So it looks a lot like the WDVAX I first showed you, and that is highly porous. What's different about this one, though, is that it in essence has shape memory properties. We can load it into a syringe, we can inject it through a needle, it will be compacted, and when it comes out the other side, it will resume its original size and shape. So with this approach, we can have a predefined architecture that we can inject, and then it readily will reform once it's in the body. Now, the first step that Nisarg did is he decided to look at how effective is vaccination in concert with the standard of care, which is chemotherapy. And so for an antigen here, he chose a particular antigen that's derived from Wilms tumor protein, which is a transcription factor that's overexpressed in these cancers. And a particular peptide from it actually has been widely explored unsuccessfully to date for vaccination. And so he did a real simple study where he did the standard chemotherapy regime, and he combined it with then providing this therapeutic vaccination. And the really striking thing he found is that when you do chemotherapy only in these animals, similar to patients, you have a really robust response, but one that goes away because the animals all relapse and they all go on to die. But if you combine chemotherapy plus the therapeutic vaccination, he got the cancer to regress, and it stayed regress actually for the rest of these animals' lives. So you get a really highly effective response. But now, to go back to this idea of, do we need antigens? Well, when you use chemotherapy and other types of cancer therapy, some of these can lead to a mode of cancer cell death that's called immunogenic cell death, which I won't get into all the details here, but it means when the cells die, they die in such a way that they can invoke really robust immune responses. And actually, the particular chemotherapy agents that we're using in the study of Nisarg's is one of these agents that can lead to immunogenic cell death. And actually, if you treat these cancer cells outside the body with this chemotherapy, you see dramatic upregulation of, in this case, Annexin-5 on the surface of these cells, which is an indication that they're dying by this immunogenic cell death. So what Nassar proposed is that maybe what we could do is we could treat animals, and someday hopefully people, with chemotherapy, induce the cells to die in a way that makes it optimal to generate an immune response, and combine that with one of our vaccines that doesn't have antigen, and let the antigen be generated at the tumor. So with this, then, you could have a personalized immune response, a personalized vaccine, without requiring personalized manufacturing. So he went ahead and did this study, and he found that indeed this actually can be a highly effective approach. We're here, you're again looking at survival in one of these animal models. The untreated animals basically go on to die quickly. If we do the chemotherapy alone, which is the ICT, you kill a lot of the cancer cells, but they rebound. And this is actually the a way of quantifying the total tumor burden in these animals. But if we gave the vaccine with antigen, all the animals were cured. And really strikingly, the bottom line, if we give the vaccine without antigen, it's just as successful, and we cure all these animals. And this just actually gives you the survival data for this study. So it looks like we're able to then generate really robust responses without even needing to identify the antigen in at least some settings. Now the mechanism here, I won't go through the details, but at least in part seems to involve the fact that we're actually attracting in some of these cancer cells into the vaccine site. They're drawn in from the GM-CSF, so when they die, they're actually presenting antigen right where we're concentrating all the antigen-presenting cells. Okay. Now, the second thing I'd like to tell you a little bit about that we're working on more recently is everything we're doing depends on being able to recruit cells, manipulate cells, have them go places, but we're basically operating blind, in that we really don't have an opportunity to follow these cells and understand if they're doing exactly what we hope they're doing. I mean, we occasionally can do studies where we euthanize animals at specific time points, and we can kind of see if they're there, but we can't really follow in any real fidelity these processes. So at Hua Wang, a current postdoctoral fellow in the lab, has proposed that maybe we could actually come up with a strategy to label these cells when they're at our materials, and then we'd be able to track them as they move around the rest of the body. And the basic premise that he's been pursuing is the idea that once we have the cells inside the device, we could have nanoparticles that we could, on demand, typically with ultrasound, release from the scaffold. These would then come out, the cells could pick them up, they could metabolize these particles and get labeled in a manner that enables us then to follow up and keep seeing these cells in the future. Now, the way he's pursued this is to do metabolic labeling with unnatural sugars. And I won't go through all the chemistry here, but we basically have sugar units that have click chemistry functionality in a nanoparticle form. And the idea is that when the cells take these up, they'll metabolize the sugars, and they'll end up on their glycocalyx, or their coating on the outside of the cell. They'll incorporate some of these unnatural sugars that now gives us a unique way of specifically going and identifying those cells later. Now, just to give you a sense of how this works, he has demonstrated that we can label the cells at the vaccine site. This is just some data where we're looking at the number of cells that have been labeled that are in the vaccine site at a particular point in time. And you can appreciate that if we have the sugar particles, so the cells can get labeled, if we release them using ultrasound, which is US, and we have the GM-CSF, the drug that makes them all concentrate, we get a lot of these labeled cells at the site of the vaccine. The other control conditions, we have very few, as you would expect. We can then track these cells as they move around the body. So here's some pretty pictures that show you that we have dendritic cells that are metabolically labeled in the draining lymph node, and then you see some quantification of that for the various conditions. So now we have an opportunity where we can start to follow these cells. We know they were once at our system, and now we can see where they go and who they interact with. So we think this could be potentially very useful from a basic science perspective to understand these systems. But the other part of this is actually if we have these cells labeled, it now gives us the opportunity to not just follow them, but to actually go ahead and manipulate them further in the body. Because the premise is we now have this click chemistry group on the cells, and we can come with the complementary click functional groups on whatever molecules we're interested in delivering to these cells and manipulating them with. So we've actually begun to explore this. And in this case, this data is just looking at delivery of a fluorophore to these cells. And there's a certain baseline level of background fluorescence that you can see with all those bars. And the really key one here is if we look at the draining lymph node, which is DLN, which is where these cells we know go, you can see that we have a really robust increase in the fluorescent signal at that site. So we are able to target, in this case, model molecules to these cells. Hua has shown that not only can we target model molecules, but we can actually deliver agents that manipulate their biology. In this case, we could deliver antigens, we could deliver adjuvants to the cells, exactly when we'd like them to get exposed to these molecules. And this enables us to take, in this case, what is otherwise a really weak vaccine system and make it a little bit stronger. And we can start to deliver things like cytokines, which cytokines are really powerful signaling molecules that manipulate a lot of the interactions of different immune cell populations. Now, normally these antigen-presenting cells secrete certain of these cytokines that then act, for example, on T cells. But then we're kind of dependent on what they're making. And what Hua is investigating now is instead the idea, well, after we have these cells generated and they're in the body, and at the time we think would be appropriate, let's introduce some of these cytokines systemically, let them bind to the cells, be concentrated locally, and then we can use these to very specifically modulate the interaction of these different immune cell populations to try to modulate the kind of immune response that we might see. So I think this actually will be a very powerful strategy that will allow us to do a lot of chemistry back in the body, as well as these kind of material-based manipulations that I've been describing to this point in time. And actually, there's not a timer up there. How am I doing for time? Do you know? Am I doing pretty well? Okay, I assume I'm doing okay. You can tell me when it's time. You can stand up when it's time for me to stop, and I'll quickly finish up. 10 minutes? Okay, perfect. Okay, that's around what I need. So I'd like to then finish up and take the last 10 minutes or so talking about another strategy we're pursuing in cancer immunotherapy using materials. And this is based on an observation that's been made lately in terms of the effectiveness of something called adaptive T-cell transfer. And the basic premise of this strategy is instead of trying to generate T-cell responses in the body, let's just instead transplant a whole boatload of T-cells to a patient, and then let this whole process be triggered. Now, this actually has already demonstrated really remarkable success. There's a particular kind of this therapy called CAR T cells, and these are genetically modified cells. And what has been demonstrated that when patients with otherwise incurable types of disease, blood cancers, are treated, you see remarkable clearing of the tumors. So what this image shows you is a particular patient, one of the first patients to be treated with these CAR T cells. And on the left, all the little black areas throughout the body are the cancer. And you can see how after the CAR T cell therapy, the cancer's been cleared from large regions of the body. And this has led to actually over 80% remission in a number of different kinds of, or in a couple of different kinds of cancer, has led to the first FDA-approved genetically modified cells as therapeutics, and has generated a tremendous amount of excitement. Actually, at this point in time, there's over 700, in addition to the FDA-approved products, there's only over 750 CAR T therapies under development for cancer alone. So there's simply an explosion of interest and activity in the idea of using these modified T cells. Now, we became interested in this mainly from the perspective of, how do we actually make these cells? And if we think about it, the process is you have to isolate cells from a patient. So you isolate, 100,000 cells, 1 million T cells from a patient. These cells get activated, which is something I was describing before. They get genetically modified so that they're able to recognize the cancerous cells. And then you actually have to expand these cells to make a lot of them. And you typically reinfuse about 10 to the 8th to 10 to the 9th cells per patient. Now, this whole process ends up taking about 3 weeks. About half the time is actually just generating enough cells. So that's a challenge. A lot of the cells that you manufacture are not functional by a number of different metrics. And so while this approach has been successful, there are some really significant challenges. It's incredibly expensive for patients. We have very poor control, for example, over toxicity, probably because we don't have very good control over the cells that we actually deliver to a patient. And the timing, it takes a long time. And right now, about 10% of the patients actually die before they can get their own cells back because it just takes too long. So we became interested in saying, could we actually improve this process? And to start with, we looked at the processes that are currently used to manufacture T cells. And there's basically 2 flavors of these. The first flavor will now be familiar with you after the earlier part of the talk, which is you take these T cells and you also isolate these antigen-presenting cells from the same patient. and you use, that's the bottom panel there, and so you use these monocyte-derived dendritic cells to stimulate the T cells outside the body and generate lots of these. Now this can work, but now you're isolating two different cell populations from patients. If the genetics of the patient are leading them to this disease, well then maybe their cells aren't that functional anymore. And so that just is a system that's very hard to scale or actually really do clinically. So instead what's been pursued is to create what's called an artificial antigen-presenting cell, which is basically particles, and the lead approach is polystyrene beads called dynabeads, to which you've conjugated some of the cues that stimulate T cell and make them active. Now, if you think about this, is a very unphysiologic way of presenting these cues. They're not presented from a cell membrane that's fluid and allows the cells to remodel and rearrange them. At the end of the day, you have to remove these particles and you have suboptimal activation. So based on this, what we proposed is that we could create a better type of artificial antigen-presenting cell that would more closely mimic the biology that normally happens between these two cell types. And the basic premise here is we start actually with the same type of mesoporous silica rods that I described earlier. And we can absorb cytokines to these and have them be released in a sustained manner, which would be very similar to the way actually then that these would be released by the normal antigen-presenting cells, except here we can control which peptide. So this is just an image of what these particles look like. We can then coat them with liposomes to create a supported lipid bilayer that would look a lot like a cell membrane. And this is just a FRAP study showing you that if we photo-bleach part of one of these particles, you lose the fluorescence and it's rapidly regained, which means that the lipids that are fluorescently labeled around that area can rapidly diffuse in. So we do have a lipid-like membrane there. We can then look over time for the release of these bioactive agents, and we're typically looking for about a week of release, continuous release of these cytokines. Now, unlike the normal cells, here we can put the exact quantity of stimulatory cues that we want, and we can vary it to try to tune the resulting biological response, and this just shows you how we can vary the amount of antibody we put on these molecules. And now when we combine these with cells, the T cells are red here, and the particles are green, and you can see the cells then start to adhere to these particles, cluster them together, and form little balls of cells and particles. And this work was all done by Alex Chung on your left, who just finished up in the lab recently, and David on the right here, David Zhang, who's currently a Ph.D. student in the laboratory. Now, what the surface of these particles looks like is shown schematically here, and this is for what's called a polyclonal T-cell expansion, where we have our fluid membrane. We have the ability to conjugate antibodies that in this case bind to the T-cell receptor on the T-cell, as well as another receptor that we know is important, the CD28, and we can release these IL-2 cytokines. And the question becomes, how does it work? So if we take human T cells and put them into these systems and compare how much expansion we generate, what you can appreciate is the dynabeads works pretty well. We get about 250 times expansion of the cell number over about 14 days. With this system, though, we get about 3,500-fold expansion. So we're able to grow a lot more cells a lot more quickly. Now, we might lead to, if we're growing them more rapidly, maybe we end up with bad cells. But what the data on the right shows you is if we look at a particular surface expression of a particular molecule, it stays very low, telling us that we're not getting more cells at the cost of bad cells. Now, this is with polyclonal expansion. We can also do an antigen-specific expansion, where instead of having an antibody against a T cell receptor, we actually mimic more closely how dendritic cells engage with T cells and use something called an MHC molecule that normally presents these antigens or these peptides to the T cells. And so we can present those along with the other cues. And to explore whether this system can actually achieve this kind of expansion, we actually did this with T cells isolated from patients that we screened for exposure to particular viruses. So the biology, this is fairly well understood. So we actually know that there's T cells there that have a specific antigen specificity. And then we combine them with our system, and we look to see if we get an antigen-specific expansion. And what this data shows you is with basically two different flavors of these T cells. So we could see it wasn't just kind of a one-off thing. We get a really robust expansion in the orange circles on your left when we use this synthetic system for expansion of the cells. And it's quite striking. The rest of these are all controls, but it is important to notice that the autologous dendritic cells, so the native cells, give you a much reduced effect. So we can actually have a much more dramatic effect with the synthetic system than we can even with the natural cells. Furthermore, as I mentioned earlier, the system is really tunable, so we can control the cues that we present to the cells and vary them. And this allows us to do something what we think will be very important, perhaps even most important, is we can change the phenotype of the T cells, for those of you in this space. And we can either have more effector cells or more of different flavors of memory cells, which may be really important for long-term persistence of the therapy. So we think this is going to be a really nice tunable system for expansion of these cells. Now, at the end of the day, what matters most, though, is are these actually effective back in the body at killing cancer cells? So we've actually replicated all the work that Novartis had done to develop their CAR T therapy that was FDA approved, and we actually used the same types of CAR T cells that they've been using, and we explored in the same preclinical model, which is a mouse model in which you introduce human cancer cells. Now, when we looked at expansion of these human CAR T cells, we find that, again, this APCMS system actually leads to much more dramatic expansion of the cells than Dynabeads, which is what's used commercially. And I haven't mentioned it before, but a nice feature of these particles is that they dissolve over time, and we can design how quickly they dissolve. In this case, by 7 days, we actually can't find the particles anymore, which means we don't have to separate them at the end of the process because they're simply not there. And now if we go to this cancer model, we introduce the T cells into the animals that have the cancer cells. We can look at the fluorescence, which indicates the total amount of cancer cells, or the survival on the right. And the blue is basically the Dynabead expanded cells, and the orange is the cells that we've expanded. And so we grew a lot more cells a lot more quickly, but when we transplanted the same number, we got at least as good of a function and actually statistically better effect in terms of cancer killing ability. So it looks like we can dramatically enhance kind of the manufacturing of the cells in terms of rates and numbers, as well as tune the quality of those cells to get a better functional outcome. So we think this particular system may be broadly useful. I've given you a little flavor of how it can be used with a limited set of cues to promote polyclonal or antigen-specific T cell expansion. However, we think that this will actually be amenable to incorporating lots of different cues that we know are important in T cell biology and may enable us to expand a wide variety of other types of cells as well. And I should mention as a disclosure, we've recently licensed this technology to a small startup that's just getting going out in San Francisco called Imulus. So I'm going to wrap up here, and hopefully what I've been able to give you a little flavor of how we've been developing what I'd call cell factories that exist either inside or outside the body. And the basic premise is we want to control in time and space how cells interact with modulatory agents. And we do this by bringing the cells to the device. We think this may be a broadly useful strategy for cancer therapies, and I didn't have time to describe it today, but also a strategy to promote regeneration. And I should mention that the work that we're doing here is now actually being supported by an immunoengineering to improve immunotherapies, basically a new center that's funded by the National Cancer Institute and the National Institute of Aging that we're trying to actually further develop and translate these therapies in terms of T cell expansion, vaccine approaches, and then a project I didn't have time to talk to you about today that's part of the center, which is developing strategies to promote T cell neogenesis in the context where patients lack T cells to start with. So I'm happy to, if there's any time left, I'd be happy to take some questions.
Fantastic. Thank you so much. All right. A few minutes maybe for a couple questions here. Please seek a microphone if possible. Any questions? Right here, this one right in the center.
Yeah, so that was an amazing talk. Thank you for coming. And I was wondering about these injections. Do you need to have to inject them at a specific location? Or, for example, the ones for immunogenic cell death, do you need to know exactly where the tumors are, and like how would you address metastases that are hidden?
Yeah, actually, so for the blood cancers, actually, we're not putting them any place in particular, but those cancers are disseminated, and so that's probably why it does not matter so much that pretty much any draining lymph node to which our cells go, there's probably going to be some of the cancer cells that end up dying there as well. For the context of solid tumors, though, then it presumably is going to make a very big difference. So I didn't have time to go through that work, but we've also been doing this in the context of solid tumors, and there we actually inject the vaccine into a tumor early or paratumorally, so we're bringing the antigen-presenting cells to the place where the cancer cells are dying.
Yeah, but what about for example, if they had moved to a hidden location that you, like peripheral secondary tumors?
Yeah, so one of the premises of immunotherapy is if you're able to generate a robust immune response against a range of antigens, and that's what hopefully we're doing here, that then even though you have cancer in different locations, once you generate an effective T cell response, the T cells circulate throughout the body and can find metastases wherever else they may be.
Awesome. Thank you.
Yep. Sam? Yes, microphone is right here.
I was going to go before Sam, but go ahead.
Hey, oh, Sam, yes, go first.
Great talk. From the early part of the talk, how does the biodegradation rate of the vaccine affect efficacy? Is that an important variable? And then I have a follow-up question from the end of the talk.
Okay, so for the first one, yes, the biodegradation rate does matter. What we've found is for our strategy to be effective, we need the vaccine to stay in place for about 2 weeks. And you actually have a fairly monotonic relationship between the robustness of the immune response and how long the vaccine site stays active, which is probably a big reason why this is effective, because we're having a lot of things happen over time. After 2 weeks, it doesn't seem to really matter.
Now, from the last part of your talk, how do you isolate the cells from the scaffold?
Okay.
I mean, that doesn't seem too simple.
Oh, actually, in this case, so the particles actually dissolve.
The particles, which particles?
The particles we're using as the artificial antigen presenting cell.
Okay, so they dissolve.
So they're dissolved. So then you basically, at the end of the day, in whichever manufacturing system you're using for your T cells, you now have a population of T cells with no foreign particles.
Right.
So we typically wash.
So the number of components include, I mean, you have liposomes.
Yes.
And you have the other particles.
Mesopore silica, yep.
The silica. So there's 2 artificial particles plus the cells.
Well, there's the mesopore silica. It's coated
Yeah.
with the lipid bilayer. So there's one particle that's coated, and then that's combined with the cells. And then the silica part dissolves. Presumably the lipids then become free. We wash at the end, but don't have to do a physical separation like you do with Dynabeads.
Fantastic work. I had a question about the cytotoxin-induced antigen process, right? I know there have been some successes. How generalizable do you think that's going to be across, say, solid tumor types and across cytotoxic drugs? So
Yeah.
paclitaxel famously, but cisplatin maybe. But how generalizable is that? Is that a universal solution, or where does it fall down, I guess?
Yeah, so it's not universal because you have to be inducing cell death in the right way. So there's a few chemotactic agents or chemotherapeutic agents that are effective in this way. So in this case, we're using doxorubicin, and there's several others. Actually, radiation also is known to lead to immunogenic cell death. There's actually a whole slew of clinical trials underway right now where they're combining kind of classic radiation therapy with checkpoint to see if then, that can be useful together. So I think it's not universal, but there's a few ways of doing it. Whether it will be, as successful across the board in all types of cancer, I doubt it. I think, like everything in cancer, that it will probably be effective in some flavors and probably not other flavors. So I think there's rooms for lots of innovations and lots of different therapies.
Any other questions?
There's somebody over there.
Hi, thank you so much. Great talk. I had a question specifically about where you used chemotherapy and then presented an antigen-less system.
Yes.
Have you noticed in that case, because it's not just the cancer cells dying, releasing particular signatures, do you get any other generated immune effects against normal healthy tissues?
Yeah, so the question you're asking is a really important one, which generally speaking with all these strategies, do we generate autoimmune disease, right? So we're breaking tolerance. We know we're breaking tolerance. So are we breaking tolerance to lots of things that we don't want to break tolerance to? In that system, we've not noticed, but we've not looked in depth. In the first system I described that has translated to the clinic, there we had to do some quite extensive studies, preclinical studies to address safety. And you actually do see some breaking of tolerance to melanin. We actually get some depigmentation at the vaccination site. which has been noted before in other therapeutic vaccines. It's actually, the clinicians look at it as being a positive sign as long as it's very localized, because it means you're actually having a biological effect. But it certainly is a risk. And at this point in time, we have 25 patients of data that it's not, but to really know that, you're going to need thousands of patients to really address these types of questions.
Time for maybe one more question. It's one over there.
Hi, so I would like to ask a question about the injectable system that had shape memory.
Yes.
Can you talk a little bit about the composition of manufacturing that permits this property?
Yeah, I completely skipped that, sorry. So it's, those are cryogels. So we typically are fabricating those from hydrogel forming materials, hyaluronic acid, gelatin, alginate. We tend to use materials that have a track record of use in humans. We modify them so we can covalently cross-link those, and we do that in a partially frozen state. So that you have ice crystals and you concentrate the polymer in the liquid phase that's around these ice crystals. You induce the cross-linking, so you get this type of porous structure. And if you do it correctly in the right range of polymer concentrations and temperature regime, you get a material that actually is highly elastic. And actually the details of that are in some of the publications. So.
Great, so please allow me to thank Professor Mooney one more time. It's such a fantastic talk.